Before we dive into today's topic, let's take a moment to review what all we'll be able to learn today. First of all, we'll be able to define what is science and what is the purpose of science. Next, we'll be able to walk through the steps of scientific method and we'll understand what are the systematic way in which a scientist asks a question and tries to find the answer based on evidence. Next, we'll be able to distinguish between what is scientific from a non-scientific approach of solving a problem. Not all questions require to be answered in a lab or with a more tabular form of data, but still you can have a scientific or non-scientific approach to solving a problem. Then we'll break down what goes into an experiment and we'll learn about terms like hypotheses, variables, controls, replicates, and what blinding is. We'll also talk about certain jargons in the field of science such as rigor, reproducibility, and objectivity. Finally, we'll apply everything we learn to a real world situation and a more humorous situation because I believe that if you can find some fun in science, it's easier to learn. Now let's begin with the question, what exactly is science? So science is a systematic way of understanding the natural world. That means it's not based on some guesswork or beliefs, but we follow a certain procedure of thinking about a situation clearly and in a logical manner to find the answers to a problem. Science is based on evidence, observation, and experimentation. Scientists make observation, collect data, and then they test that idea on the basis of what circumstances they have and then analyze whether their ideas are right or wrong. The ultimate aim of science is to be able to explain natural phenomena, things that happen around us, and they should be testable and we should be able to verify our findings. Science helps us understand so many things around us. For example, how does the moon causes tides, or how is the blood flowing in your blood vessels, what leads to increase or decrease of your body temperature, how do plants produce food. So science is not only a package of knowledge and things that you have to memorize. Science is about developing an approach of answering difficult questions. So what are the goals of science? The first goal is to build an organized knowledge base. So all the data that we have around us, we don't just randomly throw facts around. We should be able to connect the dots between those facts and derive some logic out of it. Science helps us make predictions. Once we understand how something works, we can predict how it will behave under certain circumstances. For example, if you know how bacteria grows, then you can also make a hypothesis about if I use certain kind of drugs, how will it affect the growth of that bacteria. Science helps us to solve various problems. For example, discovery of drugs, development of vaccines, even building roads, creating high efficiency computers. Science helps us solve so many problems in our real life. Science helps us understand the cause and effect relationship. If we understand the mechanism of how things work, we can understand what factors, when happen, will produce what kind of effects in that system. So we can say that science is both about gaining knowledge and using that knowledge to predict, solve, and understand the world around us. So if we are trying to develop a scientific thinking in ourselves, what are the main features of thinking like a scientist or having a scientific thinking? First thing is being reflective. We should be able to examine our own thoughts and emotions and processes that enhance our understanding and practices. We should be able to keep our biases away. We should be more objective. We should not think about our previous beliefs or bias or personal opinion, but we should be able to look at the data and evidence and make analysis, conclusions, or predictions about a phenomena based on the real data that we have. We should be flexible. We should be ready to change our opinions based on new findings. We should be able to develop independent thinking and not think in association with groups, communities, or society that we come from. So it's all about leaving your beliefs behind and try to find new science or new information based on new observations, new experimentations, and data. Being observant, you can say, is the key point of being a scientist. You should be able to observe things around you, be curious about how things work, and also be excited about finding answers. And ultimately, being empathetic is also one of the scientific thinking approaches, because yes, you can find new things which disagree with the beliefs of other people, but instead of being arrogant about your own science, you should be able to make other people understand how your new data or your new findings is helping you explain things better than what they believe already. There are different types of scientific reasoning. For example, the first type is deductive reasoning. In this, you take some general observations or general rules and try to make some certain specific conclusions. For example, you make an observation that all birds can fly. So you would make a conclusion that it's a characteristic of a bird to be able to fly. In another kind of reasoning, which is inductive reasoning, you start with a very specific observation and then you make a general conclusion. For example, if you say, if birds can fly, then make a general observation that a penguin must also fly. But that's not always true, because you know that penguin can't fly. So taking something very specific and try to apply it on everything is not always possible, because exceptions are always there. So one very safe method of scientific reasoning would be abductive reasoning, in which you have some observations and then you try to make best possible prediction out of it. And then once you have those predictions, you can test those predictions, or you can perform experiments to test those predictions. For example, if you find some feathers in your backyard, you would make a prediction that there might be some birds that came to the backyard and they left their feathers there, or it gives you a prediction that birds come to your backyard. And then you can set up some experiments. For example, you can put a camera, or you can put some food and see if the food disappears after some amount of time, then there must be birds coming in that eat them. Or if you have a camera installed, you can see that birds do come to your backyard. Now, there's nothing in this world that is unlimited or limitlessly powerful. So is the case with science. Science can also explain certain things, but then cannot explain the other things. For example, what can science do? Science can explain natural causes of events. It can help us make predictions. It can help us improve technology, medicine, agriculture. It can help us develop hypotheses and help us create some methods to test those hypotheses. For example, science has helped us understand how DNA is a molecule that carries genetic information from one generation to the next. But then science is not capable of answering some different kind of questions. For example, some supernatural things, or some philosophical questions like about the existence of God, or from where has human beings come. Yes, we have a theory about evolution, how human beings might have evolved from smaller things. But then if there is a stronger belief, there are things that cannot be for certainly explained by science. Then we cannot determine the moral or ethical values we cannot say that according to science, if euthanasia or killing somebody is should be allowed, but then morally, is it a good thing to be able to let somebody die or give somebody a choice of euthanasia or killing themselves? There is nothing like an ultimate truth. So we can say that whatever science is saying is the ultimate truth, but it is the best explanation based on facts. It can also not make aesthetic judgments. Science cannot tell if something is beautiful or not, if something is tasty or not, if something is musical or not. All these things are very subjective, they are very personal. Some person can find something tasting very good while the other person would say that it didn't taste really good. So these experiences are also very personal and cannot be 100% explained by science. So science faces several limitations. One is human bias. We scientists are human beings, so we do have our own biases about, I would like to do this kind of experiment. For example, cloning. Some people would say yes, it is a good idea to create a clone of a human being, but then other would say no, it's ethically wrong to make a replica or a clone of something. Then funding and politics becomes a situation. There are some kind of research that government or politicians would like to support, or funding agencies would like to support, but then there are some questions which the government or funding agencies are not interested in investing. um, then there is problem of incomplete data or not having very precise or advanced tool in answering several questions. For example, if you want to look at a very minute structure which we haven't even started thinking about, because our thinking is limited by the tools we have. We have some sophisticated microscopes, we have electron microscopes, we are able to say that there is an atom, inside atom there are subatomic particles, but what is beyond that? What's the limit to the ultimate smallest thing or the ultimate biggest things? We just know what we know today. There are so many things beyond that. We don't have tools to study them, so we don't know about them. And continuing to this point, there are observational limitations. Because we can't see, we can't explain what is beyond this galaxy or what happened like 10 million years ago. We don't know. We can just make some predictions based on some evidences or tools that we have, but can't say for sure. Next thing is ethical boundaries, which again comes back to the point of human bias, where we say that certain kind of experiments are um, are acceptable based on my beliefs or my principles or my values, but then I would not like to do some other kind of experiments which are not uh, aligning with my values. For example, you have to test a drug in mice, and you are a kind of person who would not want to kill a mice. Then you know that if you want to test a drug, it will end up killing the mice. So it becomes a question for you, would you want to do that experiment or not? Science isn't the only way people try to understand the world. Sometimes we come across ideas that look like science but actually aren't, and they fall in the category of pseudoscience or science denial. Pseudoscience is when people want to believe in something even though they have very little or no real evidence. Things like astrology or homeopathy fall under this category. They often have a very low standard of evidence. People accept the ideas without any strong proofs. And pseudoscience is often cherry picking information that supports the belief and ignores anything that does not support what you believe. On the other hand, science denial happens when people refuse to accept the real science or scientific evidence because it goes against what they want to believe. For example, flat earthers or anti vaxxers. they do not want to believe the evidence that has been gathered over time about the earth being round, or about the vaccines being life savers. These groups often demand very high standard of proof, and so these demands of strong evidence can be so high that no amount of data or no amount of evidence that you give them, they would be ready to believe in science. So our goal as scientists is to be able to distinguish real science from not very scientific things such as pseudoscience or science denial, and be more objective, consistent, and reliable on our scientific method or scientific approach. So now it becomes important for us to understand what is a scientific method. So scientific method is a systematic process used to answer questions and test hypotheses. It involves observation, experimentation, analysis, and then evidence-based conclusions. It aims at removing our personal biases or overgeneralization of our observations. And it ensures that we are objective, we are consistent, and we should be able to reproduce our findings. However many times you gather similar information, you should be able to reach similar conclusions. So here is a flowchart that explains the steps of scientific method. You start by making some observations. Based on those observations, you have an inquisitive or curious approach towards it. You ask a question. Then you predict the answer, or predict what is the reasoning or explanation for that question. So you develop hypotheses. The hypotheses are of two types: null hypotheses and alternate hypotheses. In null hypotheses, you make a condition in which you say that there is no correlation between the two factors that you are trying to study. And in alternate hypotheses, you would predict that yes, there can be some cause or effect relationship between the two factors that you want to study. In experimentation, then you perform experiments to study what is the effect of one factor on the other. And then you gather all the results, you analyze those results, and then you say whatever predictions you made, is your data supporting that hypothesis or it's rejecting that hypothesis? If you have reached a definite conclusion, then you go on and publish your findings. And if you're not able to make any definite conclusion, or your hypothesis was wrong, then you go back to your board and design a new hypothesis and try to make new predictions, then test that prediction again, perform experiment. If your second prediction also turns out to be wrong, then you go on and make your third prediction, and you keep on making such predictions, hypotheses, and testing those hypotheses. That's why scientists sometimes become very old sitting in their labs, and that's why in movies you see scientists as old people. So let's observe the steps of a scientific method through some examples. So the first step is observation. For example, you see that your cat knocks over the same mug every time, the red mug. So that's your observation. Or a more scientific observation would be that you have two plants, and the plant that is closer to the window is growing faster than the plant that is at the corner of your room. Then you ask a question about your observation. So in cat scenario, your question would be, does your cat hate the mug? And in the plant situation, your question would be, why does the plant closer to the window grow better? Then you make a tentative prediction or hypothesis. In the case of cat, you say that the cat prefers blue mug and dislikes red mug. And in case of the plant, you would say that because the plant which is closer to the window is receiving more sunlight, and sunlight is helping it grow better. So here scientific premise comes in, that means you should have some previous knowledge, some observation, some knowledge or evidence about what you're trying to explain. Let's say, for example, you never knew about uh, the effect of sunlight on plant, then you can't really say anything about it. You would never guess that sunlight can have an effect on plant growth. But because we have so much information about this thing, that plants perform photosynthesis in the presence of sunlight, we have this kind of basic idea, some clue, and that clue is helping you develop a prediction. So the clues or some background information that you have is called the scientific premise. Then you would set up an experiment. In case of cat, you swap the red mug with blue mug and see if blue mug is the only option that cat has, will it knock it off or not? And in the case of plant, you would say that if I move the underdeveloped plant closer to the window, will that start growing better? Or there are some other factors not related to light which are hampering the growth of the plant in the corner. Now, while designing the experiment, you have to consider so many things. You have to see what variables are independent and what variables are dependent. So independent variables are things that are changing independently, and dependent variables are what you are measuring, what you're recording, and based on which you're going to make some correlations. Then you're supposed to have some controls. For example, if you only have blue and red mug for the cat, it will be hard for you to make certain conclusions. You can put different color cups around the cat to as a control to see if the cat has preference for the color or the shape or texture of the mug, or there's some other thing that's affecting its behavior. Then about replicates, you're supposed to repeat your experiment certain number of times. You're supposed to have certain number of controls and certain number of testable samples so that you can for sure make your conclusions. Blinding an experiment is when you remove your own bias and you're not selecting. For example, you think that the cat likes blue color, so you're only giving it option between blue and red, and you're deliberately removing any other color or mugs, so that will be a bias. So in an unbiased or blinding experiment, you would take yourself out of this equation and you would ask a friend to put different kinds of mugs in front of the cat, so that that person doesn't know about your biases, which is you think that the cat likes blue color more. Scientific rigor is about maintaining certain standard of your experiment, how precisely you are recording your findings, how reproducible your findings are, and how objective you have been towards your scientific method. You have to remove your bias and you have to be more objective towards your findings and recording your findings. So in case of cat experiment, your independent variable are mugs. And what's changing? What's changing is whether the cat is knocking something out or not. Your control would be some different color mug or a neutral color mug. And replicates is how many times you make those observations, or how many different kinds of mugs did you have. And blinding would be asking someone else to set up the mugs for you, and they would do as per their own choice, how random they want it to be. In a plant experiment, the independent variable is light. And what are we measuring? We are measuring the effect of light on the growth of the plant. What controls can we have? We can keep one plant in dark. So we have moved the plant that was previously in the corner in the window. So we have two plants in the window now, but then we can keep a third plant in dark and see and compare what is changing. We can have many plants, many different situations, and different lengths of time. All those are considered replicates of an experiment. And blinding would be not very necessary here, because you don't really have any bias against this kind of experiment. You're just taking the plant and you just put that plant in front of light or in the window. How you can actually make blind observations is, so let's say there is very small change in the size, and you would say that, oh, this change was meant to happen, it's not affected by light, but just a random change in the size of the plant. So you can have some statistical analysis and make sure that the change that you're seeing is statistically significant or not. So here, while collecting the data, we give the cat many different types of cups, and we see that it knocks over the red mug 9 out of 10 times. So that's a very significant number, and it never touches the blue mug. On the other hand, with the plant experiment, you see that there is a 30% increase in the growth of plant. But is that 30% increase in the growth a result of light's effect, or it was just something that was supposed to happen anyways, even if the plant was kept in dark? So here comes the role of statistical analysis. You analyze the data through some statistical tools and make sure that the observations are significant. The changes are actually related to your cause and effect. So while making your conclusions, you go back to your drawing board where you had made some hypotheses or predictions, and then you say, my hypothesis was right, or my hypothesis has been rejected, and what could have been the sources of error, how I can improve my experiment, what are the limitations of this experiment. So with cat's experiment, we actually saw that every time cat is knocking out the red mug, and it actually dislikes the red mug, that actually supports your prediction. With the plant on the other side, you see that the plant, when moved to window, had better growth, which is statistically significant, and you can say that yes, my hypothesis was correct, that sunlight does enhance the growth of plant, which is why the plant which was previously kept in the window was growing better than the plant which was in the corner. Now that you have definite results, you have definite conclusions, you can go on and publish your findings. You find an appropriate journal. For example, for cat study, you will find a journal on animal behavior, and for plant study, you would say, I'm going to publish my findings in the Journal of Plant Physiology, and then you publish your findings. But let's say your hypothesis was rejected and you don't have any definite conclusion of why cat was behaving like that or why plant was growing like that, then you go back and test more ideas which you think could be responsible for these observations. So here is a quick recap of the definitions or the jargons of scientific method that you should memorize and try to understand, and you can go through them. You can pause the video and read through them. And here we conclude our first lecture. What did we learn today? We learned that science uses evidence and experimentation to understand the world. Scientific method, or your effort of becoming a scientist with scientific thinking, would include thinking about a problem more logically, break it down into steps, test your hypotheses, and try to make certain conclusions. Hypotheses are the testable ideas based on your scientific premise, based on your background knowledge. You should have some idea about the problem, and you make a hypothesis or guesswork based on that, and then you go on to perform experiments. In a good experiment, you should have controls, variables, replicates, and sometimes it's a good idea to have blinding. That means you take the scientist away from the experiment and let someone else make the arrangements for you so that your personal bias is removed from the experimental setup. Scientific rigor is how nicely you are designing your experiment, how unbiasedly you are gathering your observations. You should have good number of replicates in the experiment. You should have blinded studies. Your observations should be recorded through the tools which are making accurate measurements, and your experiment should be reproducible. You should be able to repeat the same experiment and have similar findings. Yes, there are several limitations to science, which, because the science can be influenced by your personal bias, your judgment, the society that you live in, your moral standards, your ethical values, some supernatural or unexplainable things which are beyond the scope of science, which for which we don't even have any tools to study at this point of time. But it is always a good idea to have a scientific thinking. At least we would like to apply these in solving serious problems and also everyday problems. Like, even sometimes it's also a very good idea to have scientific thinking in your personal situations, because it helps you see things clearly, design the experiments, or try to change the situations in a way that gives you more well-defined answer or a path towards your next situation.